and the biobank that's attached to them at Vanderbilt. And so this opened up kind of a newer area of investigation for me, something that I've been interested in a long time, but hadn't had the resources to investigate. And so that's basically using EHR-based genomic approaches to try to better understand the relationship between mental health and physical health. So that's kind of the story that I'm going to be talking to you about today. Let's see if I can advance my slides. There we go. Okay, so so this relationship between mental health and physical health is kind of well known that it's important, and one of the I think nice summary statements about the importance of this comes from the World Health Organization. On their website, they state that there is no health without mental health. And it's been documented for some time that poor mental health is a risk factor for a number of physical conditions, particularly chronic conditions. And so people with severe psychiatric illness are at much higher risk of experiencing chronic physical conditions. And vice versa, people with chronic physical conditions are also at risk of declining mental health. And so I've actually come to think about this much like a disparity, in fact, that people with severe psychiatric illness experience these kinds of healthcare disparities. There we go. So this is really sort of punctuated by the observation that the lifespan for people with severe psychiatric illness, so schizophrenia, bipolar disorder, but also neuropsychiatric disorders like autism spectrum disorders and cognitive impairment, on average, the lifespan is shortened by 10 to 12 and a half years. And it's thought that this is due to many possible reasons, one being a difference in the access to healthcare, so related to employment and insurance issues here in the States. And that's certainly been shown to play a role, particularly for psychiatric illnesses such as schizophrenia, where there's a much higher rate of homelessness. But also issues related to difficulty in expressing pain. So, for example, for people who are nonverbal, there's also some suggestion that there may be altered interoception for among people with neurodevelopmental disorders. And so for anybody who's not familiar with that term, interoception is kind of your internal perception of pain, discomfort, hunger, thirst, even sort of sensing your own heartbeat. And that appears to be somewhat altered in people with developmental disorders. But it's also certainly possible that there's some increased genetic or biological risk that's actually related to the genetic risk for the psychiatric disorder itself. So imagine sort of pleiotropic mechanisms. And then, of course, increased exposure to environmental risk factors, poor diet, lack of shelter, lack of access to medication, and those kinds of things. And I think it's also important to highlight that this is really an understudied area for a couple of primary reasons, actually. One is that until fairly recently, a lot of people with severe psychiatric illness and neurodevelopmental disorders received most of their health care in an institutionalized setting, whether that was a psychiatric institution or prison or some other kind of group institutionalized setting that was separated from community-based healthcare. And so this population has been understudied in the kind of epidemiological and community-based studies. And then there's also, as I'm sure everybody on this call is probably well aware, has been kind of a historical separation between psychiatry and the rest of medicine. And so it ends up being kind of functional separation. So that refers to the fact that, often the mental health facilities are separate from the primary care facilities. You might have like a community-based mental health clinic and a community-based primary care clinic. And even at a hospital, there's often a separate psychiatric hospital, in a completely different building from the general hospital. So there's this functional and cultural and financial separation of psychiatry and medicine that I think has also contributed to this kind of healthcare disparity and the lack of research about it. Okay. And so, like many of us in this field, this is also a very, an area of personal interest for me. So this is a picture of me with my son, Dylan. Dylan is 24 years old. He has autism and severe cognitive impairment. He's nonverbal. He requires 24-hour support staff, and he lives in a community integrated group home. And so even with a whole team of people who are focused on Dylan's health, we still really struggle to get proper health care for him. And so, you know, this is, I think, an issue that is important to me personally, as it is to many families of individuals with neurodevelopmental disorders, neuropsychiatric disorders, who are, you know, getting older and aging, and we don't really know what chronic health conditions they may be at risk for. So while on the whole, this has been an understudied area, there have been a few kind of areas of focus, particularly related to cardiovascular disease and cerebrovascular disease. And in recent years, there have been some really large meta-analyses looking at the prevalence and cumulative incidence of cardiovascular disease in people with severe psychiatric illness, primarily bipolar disorder, schizophrenia, and major depression. And so this is actually a recent paper with a meta-analysis of, I think, 92 studies that had really impressive sample size. So over 3 million patients with one of those three disorders, and over 100 million controls, where they investigated the increased risk of both cardiovascular disease and cerebrovascular disease in these populations. And so basically the take home is that there is significantly increased risk for both both cerebrovascular and cardiovascular disease, and that this risk persists even after adjusting for some of the health behaviors that may be related to the incidence of disease. So things like smoking or poor diet or BMI. And so based on these kinds of epidemiological studies that are just now starting to be published, we also are interested in asking the same kinds of questions and better understanding the relationship between mental and physical health using our EHR. So in particular, we want to understand if there's some shared biology, if it's the case that poor mental health causes poor physical health, or that poor physical health causes poor mental health. And really our model is that it's going to be all of the above. But understanding, you know, what the primary risk factors are for each type of chronic disease and where there's shared biology, I think is going to be an important area of research. Another question that we have is, you know, do these relationships between mental health and physical health transcend our diagnostic boundaries? Is it really the case that it's just people with severe psychiatric illness or diagnosable neurodevelopmental disorders who are at risk, or is it the case that you know, risk is continuous and that actually across the entire spectrum of genetic risk for these traits, that there's also increased risk for these chronic health conditions? And then are there particular health conditions for which people with developmental disabilities and severe psychiatric illness are at high risk? So there's been a lot of work done on cardiovascular disease and cerebrovascular disease, but and I think actually a fair bit of work on type 2 diabetes, but outside of those kind of primary chronic health conditions, there's really not been very much. And so we really want to look phenome-wide at these relationships. And then finally, we want to see if this can help us understand the best point of intervention, and of course to identify any interventions that are typically used in healthy populations that may cause particular problems in patients with severe psychiatric illness or developmental disorders. So maybe I'll pause there for just a minute if there are any questions.
And don't forget to unmute yourself if you have one. So, I mean, I have one short question to the first slide in there. So this shortening of the lifespan for severe physical psychiatric diseases, and so the one thing that was, from my point, was it not missing on this list is like suicide, like being internal to the psychiatric disease here? Is this not the main cause for that, or?
No, it isn't actually. It's definitely a contributory cause, but actually the primary cause is chronic health conditions.
Okay.
So suicide is definitely a increased risk in this population, absolutely, but it doesn't account for.
So you mentioned also. So you mentioned already the like being facilities, being also prisons, etc. And so, is there a big? So are there comparisons between, for example, also like the US and Europe? Are there comparisons between Caucasians and other ancestries? Have you any idea here already, or?
You know, honestly, the literature is pretty scant. So I'm not sure if that's really been investigated sort of systematically and with sufficient sample size. It really hasn't been. I mean, most of the papers looking at these associations have been published, you know, from the 90s and on. So there's not the kind of, you know, 80-year body of literature like there is for, you know, cardiovascular disease in healthy populations.
Yeah.
So I guess that, yeah, the short answer is I'm not sure that it's really been systematically investigated.
Interesting.
Yeah.
Do others have some questions? If not, no. There are like 2 or 3 on the call that are not muted. So this question, if you're on the call and not muted, please mute yourself. I can't right now. Sorry for that.
So we believe that EHR-based genomic approaches are actually a great way of investigating several of these questions that we have. So the EHR, the electronic health record, allows us to investigate the relationships between phenotypes. And the biobank that we have also facilitates investigation of the genetic relationships between phenotypes. And so we can then also compare the genetic correlations to the phenotypic correlation to better understand you know, what is going on, where there are maybe environmental risk factors that contribute to phenotypic relationships and genetic risk factors that contribute to genetic relationships between traits. And it also allows us to utilize the polygenic architecture of these complex traits and develop quantitative models so that we're not necessarily relying completely on diagnostic categories, but we can look at how genetic risk as a quantitative trait is related to risk for various phenotypes. So maybe some of you have heard me talk about the Vanderbilt EHR and the biobank, but just in case you haven't, it can be kind of thought of as 3 entities. We have what we refer to as the synthetic derivative, which is this de-identified and continuously updated mirror image of the EHR or EMR that as of now has a little over 2.8 million individuals. If we look across just that set of 2.8 million individuals, the median length of the EHR is only about a year, even though the EHR has been in existence now for 20 years. And part of the reason for this is that we're a tertiary care center. So we get people coming in from all over the state of Tennessee and Kentucky and sort of all over the Southeast, particularly, if they have if they have the need to come see a specialist in a specialty care clinic. And so we end up drawing pretty sick people from all across the state. So in comparison to like the UK Biobank that has maybe a healthier on average population, I think at Vanderbilt we have a sicker on average population. That said, there is also a population of people who make Vanderbilt their medical home, so to speak, and they get most of their primary care at Vanderbilt as well. And so our biobank, which consists of DNA samples that have been collected from just routine clinical blood draws, is enriched for that population of people that actually make Vanderbilt their medical home. And so this is illustrated by the median length of the EHR for our BioView subjects, which is about 10 years. And so we have now somewhere around 270,000 DNA samples that have been collected, and a little over 50,000 of those subjects have been genotyped with some kind of GWAS platform. And on average, the age of those subjects is around 58 years old, but we are trying to genotype and accumulate pediatric samples as well. So any quick questions about the structure of the BioView, biobank, or EHR? It's kind of central to the rest of the presentation.
So this is like all different kinds of diseases, right? So are these all cases? Are there also healthy subjects here, or is this all cases?
Well, I mean, yeah. So there are people who don't have chronic diseases, certainly, and there are people who come in just for routine healthcare, and they're in the biobank as well. So there's no ascertainment at the biobank level. That said, it's a hospital ascertained population. So I think probably most people will be a case for something. Okay,
and so how many of these 50,000 are psychiatric patients?
How many of the 50,000 have psychiatric codes? I'm not exactly sure. Yeah, I actually don't have access yet to all 50,000 samples. It's not enriched for psychiatric codes. That but we've the genotyping data is actually it's a large project. Eventually, we'll have 100,000 people genotyped, and so the data is coming through in waves, and we actually were involved in pushing several of the psychiatric diagnoses through, but we haven't gotten those genotype samples yet. So,
where are they genotyped right now?
Where are they being genotyped?
Yes.
Here at Vanderbilt.
Vanderbilt. On GSA, then the most...
No, on the Mega platform.
Okay. Great.
Okay. So as I mentioned before, we're very interested in kind of taking advantage of the fact that most complex traits have a complex genetic architecture with a measurable polygenic component. And so we can look at how the polygenic risk for all kinds of psychiatric disorders is also related to disease status for other chronic health conditions. So I think probably everybody on the call is familiar with this approach, but just in case, one of the methods that we're using is basically to calculate polygenic risk scores for everybody in our biobank. So using some kind of large discovery GWAS and taking the effect sizes from from that GWAS to create a linear weighted sum of the number of risk alleles, and then looking at how those polygenic risk scores segregate cases from controls across a number of different phenotypes phenome-wide. So we started with actually investigating this in both psychiatric disorders and in some of the previously published chronic health conditions. And so I'm starting with showing you the coronary artery disease polygenic risk scores that are significantly associated with the EHR-based definition for coronary artery disease. So here we took the beta weights from the Cardiogram plus C4D consortium study that had about 60,000 cases and 123,000 controls, and applied it to a small subset of our megadata target sample, just to make sure that indeed coronary artery disease in carefully ascertained research samples was genetically correlated to the EHR-based diagnosis for coronary artery disease. And so this included covariates, median age across the EHR, sex, the top 10 PCs, and actually it's not listed here, but also genotyping batch. And so you can see that our polygenic risk score accounted for almost 3% of the variance in CAD diagnosis within our EHR. So this was encouraging. And we actually applied this model also to the lipid traits that have been studied by the Global Lipids Consortium, so HDL, LDL, triglycerides, and tried to also model the known relationships between those risk factors and coronary artery disease. Oops. So I'm going to take just a short methodological detour here, because one of the other things that's kind of come to my attention in working with the Department for Biomedical Informatics, often when I present polygenic analyses, I get the question from people who do a lot of machine learning, like, where is your feature selection step, right? So in your genome-wide association study, you're training the weights, but then how do you know which SNPs to include in your model in the target data? And typically, really what we often do is just look across a number of thresholds and see how the R-squared might change if we include just the genome-wide significant SNPs, or everything at a p-value of less than 0.5 or less than 1, and so kind of investigate across different thresholds. But we wondered really how well we would do if we actually took a training set and used that to select a threshold for including SNPs in the polygenic risk score, and then applied it to a validation set. Because this is actually, I think, ultimately what we would do if we were looking at clinical prediction. And so really just kind of out of curiosity, we, looked at that to see how well we do now. And so this table is a little busy, but I'll walk you through it. So we have our discovery GWAS phenotype in the first column and the target phenotype in the second column. So the discovery GWAS either was the Global Lipids Consortium for the HDL, LDL, and triglycerides. Can you guys see my pointer here?
We definitely can, but there's some noise in the background. I'm not sure if this is one person that is not muted or if it's you, Leah. I'm not sure.
No, it's not me. I heard it too. That's okay.
But we see your cursor, yes.
Okay, all right, great. And so, right, so for these lipid traits, this was global lipids and for the CAD traits, or for CAD, this was the cardiogram study. And then our target phenotypes were all measured in the biobank. And so we set up a training sample of about 9,000 people, and a validation sample that had about 16,000.
Wow. Hey. Oh, hey, how's it going?
Can I think somebody needs to mute.
Jerome, If you hear us, please mute yourself.
Huh? I'm gonna present something or?
Yeah. I'm in dynamical today.
Oh, right.
And then I can
Okay.
Sorry, I don't have. I claimed the power.
Oh, cool.
Somehow they cannot hear us.
Yeah. All right. So we had a training sample of about 9,000 people, a validation sample of about 16,000 people, and so you can see the p-value threshold that was the best fit in the training sample, the number of SNPs that was included in that best fit, the R-squared value for the proportion of variance.
I have to say it's hard.
And the p-value for that.
I know. I mean, because I have no experience like neuroscience, something like that.
Sorry, it's hard.
The most would basically like channels.
So
I'm not sure. I mean, they seem to be like they don't have the speaker on, probably. They don't even listen, so it's a little bit annoying. So please, everybody, again here. So if you hear us, please mute yourself, or just if you don't listen, if you're not listening, just leave the call. That's fine as well. I don't think they. Okay.
Okay. So we use the same threshold that was identified in the training sample to define the PRS in the validation sample. You'll notice there's a different number of SNPs included. And we think that this is because the training sample was a Omni platform and the validation sample was the Mega platform. Even though they were both imputed to the same reference panel, and they're both European populations, the Mega sample seems to have better overlap with the original discovery GWAS. And so the R-squareds for both the training and validation sample, particularly for the lipid traits, are really pretty impressive and actually start to approach the SNP-based heritability for, in particular for HDL. And so I've put an asterisk here for the traits where the p-value threshold in the training sample was actually the same as the best fit p-value threshold in the validation sample. So I can pause there for any questions. We were actually kind of surprised that there were any traits where the best fit threshold was the same across multiple samples. And so I think this is actually really interesting and potentially indicating that we're starting to approach kind of maximum information for HDL and LDL in our GWAS.
That is an interesting final project for you guys. And one topic.
So, all right, in the interest of time, I'll just move on. So we took the same best fit thresholds and the polygenic scores developed from them and applied them in a GWAS analysis. So what you're looking at here is a Manhattan plot from our GWAS, where along the x-axis we have kind of classified sets of phenotypes, and along the y-axis the minus log 10 of the p-value for the PRS term. The direction of the arrow indicates the direction of risk. So if there were higher polygenic risk scores among cases, you'll see an up arrow versus controls, you'll see a down arrow. And so this GWAS was for for HDL. And so we see a protective effect of HDL on type 2 diabetes, and this has been previously shown. So this was a good proof of concept. We did a similar type of analysis for our LDL polygenic risk score, where again we see a strong association with dyslipidemias and with coronary artery disease. And so these are also known associations. Interestingly, so I don't have it here in this presentation, but there's the phenotypic correlations between LDL and the the diseases tested in our phenome-wide analysis were much stronger. So we saw lots of different phenotypes associated with the measured LDL, but these are the only phenotypes that are associated with genetically predicted LDL. So getting into our primary interest, which is the genetic risk for psychiatric disorders associated with diseases across the phenome, we asked whether genetic risk for MDD was associated with heart disease codes and actually all phenome-wide codes. And so we took the most recent 2018 MDD meta-analysis results that are posted on the website and did the same kind of thing where we calculated a genetic risk score in all of our 16,000 people in the mega sample. And we see really strong associations with mood disorders and depression, which we expect, associations with bipolar disorder and anxiety disorders. But then we also see some of these cardiovascular traits rising in significance as well. And so we see an association with nonspecific chest pain, which is really kind of a catch-all, as it states, nonspecific code. But was definitely interesting to us. And so we wanted to kind of investigate this a little further. And we asked whether we saw similar associations for related mental health traits. So related to major depression is also an individual's perception of loneliness. And this is something that we've been working on with a consortium group now, Abe Palmer, Dorit Boomsma, and myself, and others. We call ourselves the Lonely Consortium, and we've amassed almost 500,000 samples. And I think those of you on the MDD call have heard some reports of this already. And so we looked at the relationship between polygenic risk scores for loneliness and phenome-wide associations as well. And so this is our Manhattan plot for our loneliness GWAS. And you can see also that we are observing some enrichment of gene expression for our loneliness loci within tissues that we expect to see some enrichment. So this is in brain tissues in particular. And we also have observed several genetic correlations, and I just have a few posted here, but genetic correlations with phenotypes that are associated with sort of poor mental health, including general tiredness, lower self-rated health, and coronary artery disease. And one of the reasons that we were really interested in actually looking at the genetic relationship between loneliness and these other traits is that loneliness has in and of itself been identified as a risk factor for increased morbidity and mortality. And so there have been several kind of epidemiological studies looking at the temporal relationship between a person's self-reported loneliness and later health consequences. And so often a causal mechanism is inferred from that temporal relationship. But I think that's a little tricky because, of course, by the time somebody actually has a heart attack or has a diagnosis of coronary artery disease, that disease has been developing for many years. And so having that temporal relationship is not always an indicator of a cause and effect relationship. So a graduate student in my lab, Julia Seelock, has kind of led the effort on this work, where she looked at, again, the innate propensity to loneliness of polygenic risk scores for loneliness to see whether they were associated also with poor health outcomes. And so in this case, because we didn't have loneliness measured anywhere in our biobank, we weren't able to do this kind of like training on a separate sample for a best fit. So we just took everything at a p-value of less than 1. And even with that, we actually see a strong association with mood disorders and depression, with tobacco use disorders, but then also you can see with a whole host of coronary artery disease type phenotypes. And so this is also getting at that question that we had about the relationship between sort of dimensional traits and diagnosis itself. So again, these are not necessarily, so the sample is not enriched for major depression, and we're not looking at a diagnosis of major depression or a diagnosis of chronic loneliness. We're just looking at the genetic risk factors. So we actually, one of the, I think, benefits of having this type of data as opposed to just looking at genetic correlations with summary stats is that we can do a lot of conditional and sensitivity analyses to try to tease out some of the relationships. And so we looked at how BMI and diagnosis of major depression might influence associations. And so when we adjust for BMI, definitely we see an attenuation of the signal for coronary artery disease, although we do still see some of these phenotypes rising above phenome-wide significance. And we also see, again, a strong association with mood disorders, depression, and tobacco use disorders. When we adjust for the diagnosis of major depression, we still observe a significant association with obesity, and even though our coronary artery disease codes kind of fall below phenome-wide significance, they're still, of course, enriched among our results. And so this was actually a really important analysis because it's also well known that after having a coronary event, people become much more susceptible to a major depression episode. And so, so we wanted to make sure that our associations weren't completely driven by the major depression that may be diagnosed after the fact. And while we do see definitely an attenuation of the signal, I think with increased sample size, these associations will probably still remain phenome-wide significant. We also were interested in seeing if there was a difference between males and females. And so we stratified our PheWAS sample and looked separately. And while we do see some qualitative differences, so in females, definitely the depression and mood disorder codes remain phenome-wide significant, and in males, the MI and atherosclerosis codes remain phenome-wide significant. These differences between them were actually not statistically significant. So I think this is really just reflecting the fact that more males have myocardial infarctions and more females are diagnosed with depression. Stefan, how am I doing on time?
It depends on how many slides you have left. But,
well, what time is it?
We have still another 17 minutes, but let's leave another 5 minutes to the end at least. So you have a good 10 to 12 minutes left.
Okay, all right. Okay, great. So since we were really primarily interested in following up the associations between polygenic risk for loneliness and the coronary artery disease codes, we focused in on the males and looked there at association after adjusting for, again, MDD or BMI. And so, again, we see that myocardial infarction remains significantly associated after adjusting for either BMI or MDD, and actually also remains associated after adjusting for both MDD and BMI. And so this now, I think, is a really nice substrate for a Mendelian randomization analysis as well. So the second part here is actually just kind of an introduction to some of the work that we have planned. And so I just wanted to sort of briefly go over it and invite any ideas or collaborations if people are particularly interested in certain biomarkers for phenotypes that they are studying. So within the EHR, we have access also to actually thousands of labs, but many of those are kind of unique special snowflakes. So they may only have been ordered on a small handful of patients. So when we start looking at labs that have a larger sample size. It turns out that we have about 350 labs with over 1,000 individuals. And actually, I should say that all of these labs have at least 1,000 observations. So if we say that, we've got 350 labs with at least 1,000 individuals, at least 1,000 observations, then everybody has been measured at least once. But then we've got a larger number of labs where we have a smaller number of individuals, but a larger number of observations per individual. And so this data is also, really rich for looking at longitudinal associations between the relationship with, psychiatric illness and changes over time in various biomarkers. So in total, we've got about 500 labs with at least 100 people measured, at least 1,000 observations. So this work has, this data source really has not been utilized very much in the EHR space, partially because it's really messy data. So it's taken us close to 2 years actually to really carefully QC all of the labs that had sufficient sample size. It's also challenging because, again, it's a hospital population. And so, a lot of times the labs are being drawn because somebody is actually sick. And so this can be a challenge to interpretation. But at the same time, the fact that we have the entire EHR allows us to investigate the relationship between diagnoses and changes in lab values. Some of the benefits of using this data are that we have a really large sample size that is really rich for clinical data. So it's longitudinal. We've got over 20 years worth of data. And we can, as I've mentioned a couple of times now, test the effect of many possible mediating and moderating variables. And then we can also go into the charts themselves and validate by chart review. So developing tools to both QC and visualize this data has been a really tremendous effort by Peter Straub, a programmer in my lab. So he's developed a Shiny app and a whole set of tools that actually we're planning on making publicly available, and we'll be able to make the summary data for all of these different labs available so that we can look at how they vary by age, by sex, by race, and that groups with biobanks and labs, locally can also download these tools and apply them to their data. And this is sort of the concept map for what we would eventually like to do. So this is, that we would take the, beta values from GWAS of many psychiatric illnesses, calculate polygenic risk scores across everybody in BioView, and then look at how those risk scores are related to median values in hundreds of routinely collected labs. And as I mentioned before, we can also use longitudinal models to see how they're related to change in lab values over time. So we've started doing this a little bit for some of these traits that I've been talking about so far. So this is just focusing in again on HDL, LDL, and triglycerides, and looking at how the genetic risk scores for coronary artery disease, loneliness, or major depression is associated with median lab values. And so in each of these plots, we're looking at the R-squared values, the proportion of variance explained in HDL, LDL, and triglycerides on the y-axis, and then the discovery GWAS p-value threshold on the x-axis, so that we can see kind of across the board how well do MDD risk scores, CAD risk scores, and loneliness risk scores predict HDL, LDL, and triglycerides. And so interestingly, we see that for HDL, our loneliness risk scores actually tend to outperform our CAD risk scores. And for LDL, it looks like there's some difference, but I don't think these are actually really meaningful, because below 0.1% variance, it's not a significant association with between the risk score itself and the median value. And then in our triglyceride analysis, we do see the CAD risk score outperforming the loneliness risk score, which is kind of what we would, maybe intuitively expect. But the loneliness risk score does actually significantly predict triglyceride levels as well. So, like I've said a couple of times, we also are interested in testing for the mediating effect of these quantitative traits and the moderating effects of sex and medications and other diagnoses on these relationships as well. So this is kind of our an example of a general model that we're interested in testing. The relationship between polygenic liability and disease diagnosis that may be, again, mediated through quantitative traits. So kind of our future interests are to mine this lab data for biomarkers for neuropsychiatric disorders using genetic risk scores from our publicly available GWAS. And I should say that, we don't think it's likely that we will identify a biomarker for, major depression out of all of these routinely collected labs, but that actually we may identify several quantitative traits that together may be predictive of an individual's genetic risk for major depression or schizophrenia, for example. We also are really excited about using bidirectional Mendelian randomization analyses to better understand some of these possible causal mechanisms and to compare the phenotypic correlations with the genetic correlations to try to identify comorbidities that may actually be more of a consequence of environment than of genetic causes, which we're also equally interested in understanding. So I think that is it. I'll wrap up with an acknowledgement of everybody in my lab, a really great group of people to work with and wonderful students, and the work of the Lonely Consortium. Again, this has been like just a phenomenal collaboration that I think has yielded some really interesting results. And so I think with that, I'll end, and hopefully there's some time for questions.
Thanks so much, Leah. Fabulous presentation. It's a lot of data that's coming through there through Vanderbilt. It's really exciting to see that you're diving into that with full speed here. So is there, we have like around 30 people on the call. They probably have questions and they don't know how to unmute themselves, or I still have questions, but I don't want to always step in here. So, did I hear somebody? Not yet. So, okay. So then, Leah, I still have like 2 or 3 like more short questions, like more a little bit on the technical side. So 2 things here. So first of all, these are you able, because you're speaking about like longitudinal stuff that you can apply here, because you have like multiple measurements on the same individuals in here, but also is there a chance to actually, given the fact you might have seen some people with especially high polygenic risk scores for schizophrenia or so, is there a chance to get in contact with them again on the next visit or so? Or is this something that's so anonymized that there's no chance to actually get in contact with these individuals again?
No, it's a completely de-identified data set. Yeah. There is also what's called the research derivative. So we work within the synthetic derivative because we're also working with the genetic data. If we were to restrict ourselves just to the phenotype associations, we could apply for access to the research derivative, which is an identified data set. But even then, I think there are rules. I don't know that we could actually recontact patients. And there's no, although I don't understand why this is the case, you can't work with genetic data in the identified environment.
Mm-hmm. Is this special to Vanderbilt, or is this generally for the US? Because
well, I think most of the biobanks within the US are de-identified. That issue about not working with genetic data in the context of an identified environment, that might be specific to Vanderbilt. I'm not actually sure what the rules around that, where those came from.
Very interesting, yeah, and a little bit sad, of course, because that would be then the golden example, right?
Yeah.
And so the other issue, a little bit more on the technical side, I mean, we all know that, like, especially polygenic risk scores are relatively sensitive to sample overlap. Mm-hmm. So how is there any chance for you, or how do you want, how are you dealing with this issue, like?
Yeah. No, that's a good question, and we've been thinking about this a lot. So, so there's a method, I can't remember now the first author's name, but I think it came from Peter Vischer's group, in the context that we're using, where we have some individual level data and GWAS summary statistics. They've published an approach to actually first just check to see if there is, what the probability of overlap actually is. Everything up until now, I have to be honest, is based on just the knowledge that no investigators have contributed actively from Vanderbilt to these GWAS. But I mean, we haven't yet done the careful checking to make sure that there's no sample overlap. But that is on our list.
Yeah, exactly. I think that's something we need to think about, especially.
Yeah, absolutely.
Would we agree?
Yeah, it's really something that I think we have to just actually bake into the pipeline. Mm. And we're also part of a collaboration with other biobank sites through the Emerge network, where we're doing a similar kind of thing, replicating these polygenic PHWAS associations in other biobanks. And so, so we're really trying to develop a robust pipeline for doing that across several EHR environments. And one of the things we need to make sure to build into the pipeline is this kind of checking.
Yeah, I mean, especially within PGC, we could, for just testing reasons, we could just try to see if we see some unexpected overlap there. And this, if we don't, then this gives you a little bit more security that there isn't. And if you suddenly, like, 200 cases coming up that were shared from somebody else to PGC, then it's obviously something that you need to be super careful with all the other phenotypes. I mean, at least in PGC, we have everything in place, and we could test that. We could test that even without, we have this checksum method where we can actually test this without sharing the genotype. So that's something that could be done on a relatively low level here.
So in a phenome-wide type of analysis, how would you then basically, like, for each PHWAS category, conduct a GWAS and check all of those SumStat relationships? Because it's also possible that we have, say, somebody with schizophrenia who doesn't actually have the diagnosis in our electronic health record, right? Yes. They have a diagnosis of type 2 diabetes in our health record, but maybe they've been included in a GWAS somewhere else. So I think, first
that's why I would be very interested to see this. Like the check on the summary statistics level works for, like, a significant overlap in there, obviously, for if you have the same phenotype, and you're raising exactly the right question. So I think if you want to be really sure, you need to really test it on a genotype level, right?
Yeah.
And then, but I think at least if you can do this like with a couple of these consortia, you at least get a feeling if there's something unexpected or something expected, and I think this gives you already some support here. And maybe when you actually see this at the summer statistics level, where you see actually some significant overlap there, I mean, you might find it somewhere, then you probably want to go there and ask these guys for, can we actually test this on a genotype level? Who are these individuals? So that this is actually correct. Is this? I think we can test this on a couple of different levels here.
Yeah. No, that's a good idea. And that's basically what we wanted to start with, was just to see first, like, do we see any evidence of overlap? And then if we do, how should we go about dealing with it?
I mean, to be honest, the overlap cannot be really, really big, because then it's so quick that you see really exploding R-squared values. This is really impressive. Even if you have a small data set, like a couple hundred individuals in our big meta-analyses, if all of these, if it's just 300 cases in there, but if all of these are in the meta-analysis, the R-squared values are exploding. So it is probably definitely, if there is something, it's definitely very small.
Yeah.
But I mean, of course, this little small thing can have this small impact on these phenotypes that seem to fit so well, right?
Yeah. So do you have an intuition of like what you expect if there's an overlap of, say, a dozen people, like a really small overlap?
No, there's always something that I really wanted to test.
Yeah.
Somebody wants to go for it, please go so. No, I don't have a clear intuition. I have these experiences that if there's like a couple hundred overlap, it's really strong. It's surprisingly strong. It's really, that's why I'm always careful about this. It's not that you see like R-squared is just jumping a little bit around. It's really exploding. And so that's why I think my hunch is that even like a dozen or 20 could have a small impact. Not big. I mean, some of your p-values are really like just passing this phenome-wide threshold there, right? So that's why, especially for these cases.
Yeah.
Okay, but let's keep this up, and if this discussion, and I'm very happy to be on there as well.
Yeah, and so for anybody on the call who's interested in looking at particular phenotypes, I mean, we've now PheWAS polygenic scores for all of the publicly available summary stats from PGC, and so anybody who's like interested in talking more about what schizophrenia looks like, or what bipolar looks like, or whatever, please feel free to get in touch with me, because I would love to kind of have more folks to discuss these with.
Fabulous. I'm sure the people will be more brave via email, I guess. Okay, so we're actually over the hour, so I will have to close the call now. Thanks so much, Leah, for presenting.
Yeah, thanks, guys.
And I hope this will continue here. So, and all the best to everybody else. See you in a short time. Bye-bye.
Bye.